Welcome to the Chapter 13 lecture on microbe and human interactions. Microbes and humans can live in close partnership, and these types of relationships between two organisms living closely together is called symbiosis. There are three types of symbiotic relationships. Commensalism is when one of the organisms in the symbiotic relationship benefits from the association and the other organism is neither harmed nor benefiting. In symbiosis that is considered mutualism, both of the organisms benefit from the close association of the two living together. And in symbiotic relationships that are considered parasitism, one of the organisms benefits and one of the organisms is harmed. Humans have microorganisms living in or on them in healthy and diseased states. When a human has microorganisms living in or on them and the human is healthy, the relationship between the human and the microorganism is either commensalism, where the microorganism would benefit and the human would be neither harmed nor benefiting, or could be mutualism, where the microorganism is benefiting from living in or on the human, and the human is benefiting from the microorganism living in or on them. In cases of disease, when a human has a microbial infection, the microbe is benefiting by living in or on the human, and the human is being harmed. So microorganisms that cause disease are called pathogens, and the relationship they have with their human host is a parasitic one. Human microbiota, which is the term used for microbes normally living in or on a healthy human, are derived from the birthing process, either in the vaginal canal or through C-section. As soon as the newborn leaves its mother's womb, it is in contact with microorganisms that are very prevalent in the environment and living everywhere. And those microorganisms will start to colonize the gut and the body of the newborn. And as the newborn feeds on breast milk formula or eventually solid foods, the composition of the microbiota in the newborn will change. Microbiota are very important for keeping humans healthy, and their presence in the human body is considered normal. In the human body, microorganisms live in nearly all of our body systems. We have plenty of microorganisms in the nose and throat, which is also called the upper respiratory system. We have microorganisms living in the conjunctiva, which is the areas around the eye. The majority of our normal microbiota is living in our large intestines, and they help us to digest our food, among other things. We have microorganisms living on our skin, and we have microorganisms living in our urinary and reproductive systems. I've listed several of the common groups of microorganisms in all of these areas, but you'll not be tested on which groups live in which part of the body. There are also some areas of the body where in a healthy human we should not see growth of microorganisms. So these are referred to as actinic, meaning free of microbes. These are the alveoli of the lung, which is the most outer portion of the lung where the blood exchange with oxygen occurs. In the blood and cardiovascular system, a healthy person should not have microorganisms growing. In the brain, in the cerebrospinal fluid, and in the uterus. The uterus, because the newborn or developing newborn does not have an immune system, so the presence of microorganisms in the uterus could be dangerous for the developing fetus. Normal microbiota are important for keeping humans healthy. One of the main reasons for this is that they protect the human host via something called microbial antagonism. And this is the idea that having normal microbiota in the body can prevent the growth of pathogenic or harmful microbiota in the body, because these normal microbiota will compete with pathogens in an ecological niche. So there's limited nutrients and space in the human body, and the presence of normal microbes will prevent the presence of pathogens in these limited spaces. Occasionally, normal microbiota can become parasitic and cause infections. When a normal microbiota that's present in a human becomes parasitic and starts causing infection, these infections are called opportunistic or endogenous infections. For a normal microbiota to become pathogenic or parasitic, something has to change in the human host. So one example is that the human host's immune system could become suppressed because they have HIV or if they've undergone an organ transplant to suppress immunity. The immune system cannot keep normal microbiota in balance, so things get off balance and normal microbiota can cause disease. Also, if the human takes antibiotic drugs, these can lead to changes in the numbers and types of normal microbiota present and could lead to disease. Or if a microbe normally grows in one area where it's not causing disease, but it changes location, in that new location it can cause disease. So an example would be Staphylococcus aureus normally grows on the surface of our skin. But if we have a wound on the skin, the Staph aureus can penetrate into the deeper tissues and cause an infection. The progress of an infection is in 5 steps, which we'll go through now. The first being a portal of entry. So a microbe that is outside of the human is called an exogenous microbe, and in order to cause infection, an exogenous microbe must first enter the human through an area that is called a portal of entry. So these are areas on the human body where microbes can get into the human body, and this would be the first step in causing an infection. So we have skin being a portal of entry, especially if it's broken or damaged by an insect bite or other injuries. We have the gastrointestinal tract being a portal of entry, so anything you swallow goes down into your gastrointestinal tract, and if you swallow a microbe, they can enter your body through that portal. The respiratory portal, which is the most common portal of entry for microorganisms, is anything that we inhale through our mouth or nose, so then enters into the lungs. So many microbes are inhaled and then get into the human host via the respiratory portal. And the urogenital portal, which is all of the sex organs and the anus. So all of the sexually transmitted infections, microbes causing those, will enter via the urogenital portal during unprotected sex. Once a microbe is in the human host, its second step in order to cause an infection would be it has to adhere to the human host tissues. So the step of adhesion can be the result of several factors on the microbe. One being if the microbe is coated in fimbriae, these can stick to the host cells. If the microbe is coated in a capsule, these also help the microbe to stick in the host cell tissues. And in viruses, we see the spikes serving this purpose, where they can adhere to host cells and help the microbe to remain attached to the host cell tissues so that it can get established and increase in numbers. Step 3 is the microbe has to be able to survive host defenses. So these would be the early defenses of the immune system. One of the first things that happens when a microbe enters a human is cells called the phagocytes will respond by trying to engulf the invading microbe and destroy it. In this image, we see a phagocyte attempting to engulf the 2 bacteria which are coated in a green capsule. Because these bacteria have a capsule, the phagocyte cannot engulf and destroy them, so the microbe therefore survives these host defenses. Other factors that can help a microbe to survive the phagocyte, which are called antiphagocytic factors, in addition to having a capsule, are leukocidins, which is molecules that microbes can release that destroy phagocytes, or some microbes can survive inside the phagocyte. So although they are engulfed by the phagocyte and normally would be destroyed, they're able to survive in the phagocyte and remain there to establish infection. Okay, step 4. After the microbes have entered, adhered, and survived host defenses, next they cause disease, which is damage to the host, by secreting molecules that are categorized as enzymes or toxins. And these molecules will cause damage to the host in some way. So several of them are listed on this slide. We have several types of toxins and enzymes on the left and right. The one being shown are toxins that would degrade connections between host cells here so that the bacteria can move into deeper tissues. Toxins are categorized as either being endotoxins or exotoxins. Endotoxins are secreted by gram-positive cells. They are made of proteins and they are released by a living bacteria. Endotoxins are toxins that are released when a gram-negative bacteria cell dies and its parts of its outer membrane are shed, and the molecule lipopolysaccharide, or LPS, is shed when the gram-negative dies, released in the host, and causes a severe immune reaction which makes the host very ill. Warning signs of disease are categorized as either symptoms or signs, where symptoms are sensed by the patient, signs are detected or measured by an observer. So when you go to the physician to be treated for an illness, the physician will ask what your symptoms are, because these are things that cannot be measured and can only be sensed by you. Examples would be nausea, malaise, fatigue, chills, and signs would be externally measurable things like fever, white blood cell count, appearance of a rash, etc. Lastly, if a microbe is able to stay in the host and establish an infection, it then will spread to new hosts via a portal of exit. So portals of exit are ways that a microbe exits one host so that it can be released into the environment to find a new host. And listed in this figure from your book are several portals of exit. You can protect portals of exit by covering your nose when you sneeze, so this would block microbes from being expelled into the air. Or another example is wearing a condom when you have sex, so the microbe can't exit out of the urogenital system. The stages of an infection are listed in this figure. So if a microbe gets established in a host and causes an infection, we have a period where there is no symptoms or signs. This is called the incubation period. And in this period, the microbe is getting established in the host. During the prodromal stage, there are mild symptoms and the host is starting to feel ill. The microbe is increasing in number inside the host. During the period of invasion, we reach the height of infections where symptoms are the worst. Microbe numbers are also likely the highest. And then when the host starts to feel better, we enter a convalescent period. In infections that can be completely cleared and the host restored to normal, we get back to a pre-disease state. But in some types of infections, like HIV, for example, where the virus is never cleared, we enter a continuation period where the host still has some symptoms, but they are largely considered cured.